Hi, everyone. There we go. Now I got my video started. Welcome. And I am hoping this is a really fun presentation for you today, talking about immune rejuvenation. As Dr. Ferris said, we have a lot to talk about today because we have gone through a huge change in immunity over the last couple of years. And before we get started, just listing some of my disclosures. None of these should be relevant to the talk today. So first of all, this is our topics. We're going to start with immune rejuvenation and what our current health status is. Then we'll talk just a little bit about stress and inflammation. We'll talk about social support, and we will end with a therapeutic approach that addresses the microbiome. I think you can probably guess what our current health status is, but let's just take a look. For today, what we're going to focus on is the fact that a viral infection has gone throughout the population. And we need to look at COVID as how we have all responded immunologically to that virus. But we can't just look at that because we also have all experienced social isolation. And when we experience social isolation, that affects our hormones and our microbial metabolites, which ultimately has an effect on our immune system as well. And we've all experienced a certain amount of stress. That might be financial stress. It might be stress of fear of infection. Regardless, that stress has an enormous impact on our immune system, our cytokines, and makes us feel poorly. So what we have to look at is how all three of these interact as we're thinking about bringing our immune system back to a state it was in 2019 before all of this happened. So let's start with a quick review of the immune response. Usually when we experience an injury or an infection, we start immediately with inflammation. And that inflammatory response are the proteins, the cytokines, and CRP and complement, and all of the things that are made in our liver that then trigger our innate immune system to kick into gear. That innate immune system are the cells like macrophages and neutrophils that are going to take up whatever infection or whatever dirt or whatever comes in through an injury. The macrophages and dendritic cells of the innate immune system then trigger the specific immune system. And this is where our T cells and our B cells are going to become involved. These are the cells that are also going to be responsible for keeping things like cancer at bay. But these cells are highly relevant to fighting the infection because some of them are going to stick around afterwards and become our immunological memory. After we mount the majority of our immune response, we need to resolve that immune response. And resolution means we have to get rid of all those cytokines and inflammatory proteins. We need to bring the cell numbers back down to normal for our innate and our specific immunity. And we need to reduce all of the inflammatory processes. Now, if we look at what normally happens every year is we have annual infections. We know that the flu and several colds circulate annually. For the average adult, you're supposed to be getting somewhere between 2 and 4 infections per year. Now, 2 to 4 infections per year, you're probably thinking, well, I don't get that many infections. This isn't something that's going to knock you down and make you go to bed for 3 weeks. An adult infection is typically a 24-hour flu. It's one of those things where you feel yourself getting sick, your immune system snaps together and nips it in the bud, and it is gone. For kids, we're used to getting 6 to 8 infections per year. And what we noticed over the past 2 years as everybody wore masks and weren't exposed to each other is most people didn't get the flu and they didn't get a cold. They didn't have any infections. The issue around no infections means that we didn't make interferon gamma. And interferon gamma is a cytokine that's really important to your immune system. If you don't make interferon gamma, then what you see is that latent infections, things that are already in your body but are not being expressed, are going to reemerge. And one of the things that we've been seeing is a reemergence of shingles, which is the varicella virus. We've also seen reemergence of Epstein-Barr virus, another virus that is latent in 95% of the population. So if we don't have interferon gamma around, all the latent infections are no longer kept in control. Another thing that interferon gamma does is it is in balance with our Th2 cytokines, with the cytokines that cause allergies. So if we don't have interferon gamma around, we make more IL-4, IL-5, and IL-13, and we have more allergies. And without interferon gamma, we also have a risk for more cancer because we know that the cytokine that's keeping cancer under control, specifically causing the induction of CD8 T cells, which can kill tumor cells, is interferon gamma. So without interferon gamma, we see other immune-based diseases emerging. Now, we also have to remember that in addition to not having our annual infections, millions of people at this point have had SARS-CoV-2. What we've seen with the immune response to SARS-CoV-2 is that we can tell a lot about the population and what's going on with their immune system based on how they respond to this virus. So we know that people who have underlying inflammation, where their inflammation starts high, it means that when they get SARS-CoV-2, that inflammation doesn't shut down at the end of the infection. It just keeps going. And furthermore, these are folks that tend to have higher levels of cytokines when they get a SARS-CoV-2 infection and risk a cytokine storm. We also know that this particular virus is really good at shutting down the innate immune system. So you have less cells that are going to trigger specific immunity. And when it comes to specific immunity, we know that virulence factors produced by the virus itself prevent specific immunity from launching in somewhere between 10 and 30% of people who get a SARS-CoV-2 infection. So some people who get this infection with the initial variant, about 30% of people did not mount a specific immune response. With the current variants, the BA.5 variants, about 10% of people are not launching a specific immune response. And the other thing that we've been seeing is that people are having very slow resolution of SARS-CoV-2. Resolution is something that is also dietary controlled. And so paying attention to the fact that people can't resolve infection is important, because we're seeing then the production of long COVID or post-COVID. We know that somewhere between 49% and 58% of people who suffer from long COVID have long-term fatigue. And you don't have to have a severe infection to have long-term fatigue. This is 49 to 58% of anyone who's had an infection, not just severe infections. We also know that 40% of survivors are having trouble breathing, and 20 to 30% of survivors are having joint pain. So the long COVID symptoms are being further elucidated all the time. This is a paper that came out a week ago. And what we see here is that there are more than 50 symptoms of long-term COVID-19. Fatigue is by far the greatest, followed by headaches and attention disorders and asomnia, or anosmia, the inability to smell. But if we look at the population of people who have had COVID-19, 80% of them, 80% of somewhere around 200 million people have at least one symptom. That's enormous. And most of these symptoms are mediated by inflammation or by the immune system. So we also know that there's potential for autoimmunity based on our immune response to COVID. For people who have the correct genetic background, if they suffer from COVID, there's both a molecular mimicry and a bystander effect that can happen. So remember, we have inflammation that doesn't shut down. So we've got our pro-inflammatory cytokines around that can trigger higher levels of specific immunity and high interferon gamma. And with our slow resolution, this is like the perfect storm for developing autoimmune disease. And in order to look at the molecular mimicry, I just wanted to show you the homology between the COVID-19 proteins and self proteins. So this is a paper that came out in January of 2021. What you'll see on the top line are the spike protein, nuclear protein, envelope protein, and membrane protein of COVID. SARS-CoV-2. So you'll see 100% reactivity because that's our positive control. Now, going down the left column, you're going to see self-antigens. And where we see high homology with self-antigens is mitochondrial antigen. So we can attack mitochondrial antigen when we're attacking viral protein, both for the spike protein, the nucleoprotein, the envelope protein, and the membrane protein. But there are some other interesting self-antigens here as well. Neurofilament protein also has high reactivity with the spike protein. Insulin receptor has high reactivity with the nucleoprotein, 45.5%. Suggesting that diabetes could be an outcome of a COVID-19 infection. Intestinal epithelial cells have high reactivity with an envelope protein, meaning that inflammatory bowel disease could be an outcome of a COVID-19 infection. And if we then put all of these things together, it suggests that autoimmunity is going to rise following COVID-19, unless we're really conscious about trying to pre-treat people for autoimmune disease. So if we look at what happens after COVID, we see post-COVID, we see inflammatory conditions, the fatigue and some of the other things. We see potential autoimmunity, and we're seeing dysbiosis. And this is not a coincidence. The immune system, 80%, is located in the gut. And we have seen two papers that show that the gut microbiota composition reflects disease severity. And we see that with disease severity, we see increase in several inflammatory cytokines, chemokines, as well as CRP and sedimentation rate. But we also see that patients with COVID-19 are depleted in particular gut bacteria, such as Faecalibacterium praenicii, Eubacterium rectali, and several Bifidobacterium species. Furthermore, and this has now been confirmed multiple times, the dysbiotic gut microbiota composition in patients with COVID-19 persists after the clearance of the virus. So we're looking at a population with dysbiosis. Now, how are you going to know if your patient has dysbiosis? Well, doctors' data happens to have a very good GI panel called GI 360, and you can order a panel like this to determine what the microbial diversity is in your patient. And you can tell whether they are a person that came out of COVID-19 with dysbiosis and whether that is something that is contributing to all of the immune sequelae that we're seeing. Remember that the gut is the immune system microbiome interface. So what we see in a healthy gut is we see a really good mucus layer, and that mucus is triggered by the commensal bacteria. So if you have dysbiosis, you're not going to get this mucosal layer, and you're going to develop a very thin layer that allows additional food proteins as well as overgrown bacteria to make it across that gut barrier. Also, as part of that mucus layer are antimicrobial peptides. These antimicrobial peptides are what are keeping normal microbiota from overgrowing. So when you don't have those, you get an overgrowth of normal commensal bacteria, which makes it pathogenic. This isn't the introduction of new pathogens. It's not like you suddenly swallowed salmonella. It's normal microbes that are now overgrowing. We also know that the short chain fatty acids that are produced by the commensal bacteria are responsible for producing all of these anti-inflammatory cytokines by our T regulatory cells. And so if we don't have those short chain fatty acids, we're also looking at a reduction in T regulatory cells. And this is what keeps autoimmunity at bay. So a lot of different pathways are involved in keeping our immune system and our gut in balance. We also know that lifestyle has a significant effect on both the immune system and the microbiome. So diet, lifestyle, stress, movement, sleep, all of those things that we didn't do for 2 years, we didn't pay attention to our diet and our lifestyle and our stress levels, are now causing this dysbiotic human. And we have to get all of these things back in alignment in order to get our healthy human with a healthy microbiome. So what does it look like? Well, according to the American Psychological Association, 42% of adults gained weight during the pandemic. They didn't just gain 10 pounds. It wasn't the freshman 15. No, the average weight gain was 29 pounds. That's average. 10% of people gained more than 50 pounds. That's enormous. And taking that weight off is going to be really complicated. 67% of Americans are either sleeping more or sleeping less since the pandemic started. What we see is around 35% are getting less sleep. That's less than 6 hours a night. And 31% are getting more sleep. And that's more than 10 hours a night. 23% report drinking more alcohol to cope with the stress. And lest you think that it is just the adults who are dealing with this, 46% of Gen Z says their lives have worsened since the pandemic. Keep in mind that these are folks that typically were graduating from college, where you would normally have the hope of going into a job and starting your career and getting really enthusiastic and excited about going to medical school. And this kind of just shut everything down. So what we're left with is that most people are going to come out of the pandemic with increased inflammation, decreased immunity to infection, activation of latent viruses, dysbiosis, and risk of autoimmunity with infection. So the past 2 years has taken a toll on our immune systems, and everyone, whether they had a COVID-19 infection or not, needs immune rejuvenation. And let's just remember how stress impacts the immune response, because stress is also increasing inflammation. If we look at stress, we have to remember that stress is a perception. We perceive a stress or a threat, and that is something that we think about, and we think, whether consciously or not, that we are inadequate to cope with whatever the situation is. When that determination is made, that activates the hypothalamus, the pituitary, and then the adrenal glands to produce corticosteroids. And the glucocorticoids are going to cause a whole body response. I put this slide in here because I want us to remember that stress is accumulated. It's not just this stressor that you might have today of, oh, I've got to squeeze another hour into my day to watch this seminar. It's accumulated. And we now are seeing that children who experience adverse childhood events, who have stress in their childhood, have additional stress later in life. And that can happen in one of two ways. It can make them either more reactive to stress or less reactive to stress. So it either stimulates a cortisol response or it blunts a cortisol response. The way that we look at that in research is by looking at allostatic load. What we're looking at is a whole body response. We recognize that stress isn't just cortisol. It also has an effect on the cardiovascular system, the metabolic system, the immune system, our behavioral health, et cetera. But the best way still today to measure stress is to look at cortisol levels. And Doctor's Data has a fantastic salivary cortisol measure. And I say it's fantastic because it's measuring saliva at multiple times a day, and we can actually look at the cortisol curve. It's not enough to take a single blood measure of cortisol. That only tells you the total level. It doesn't tell you how the cortisol is being secreted over time. And when we're looking specifically to see if a patient has a heightened stress response, if they have a higher curve, or if they have a blunted stress response where it might have flatlined, you can still have the exact same blood level, but the curve looks different. So using a salivary test, specifically the adrenal function test from Doctor's Data, will let you be able to determine whether your patient has a heightened cortisol response or a blunted response. What's happening inside the cell while this cortisol is having its actions? Well, the first thing we have to remember is that glucocorticoids have a receptor that is a transcription factor, and the glucocorticoid receptor is in the cytoplasm of the cell. When a glucocorticoid like cortisol binds to the glucocorticoid receptor, it causes translocation across the nuclear membrane through the nuclear pore into the nucleoplasm of the cell, and then it's going to sit down on the DNA and act as a transcription factor. And specifically, the genes that are modified by the glucocorticoid receptors are cytokine genes and the T cell receptor. So we make more or less protein based on whether or not we are producing glucocorticoids. Now, most of us have heard that glucocorticoids are immunosuppressive, but does all immunity cease during high stress? The answer is no. We evolved for high stress to protect us from the running from a lion or from battle. And so what happens in a high stress situation is that the immune system relocates. It's not in the gut. It's not in the lymph nodes. It goes to the skin. And what we see is that it's going to be highly dependent on the cell type, where it's initially located, the location of the stressor, the time of day, because is it when cortisol is peaking or is it when cortisol is low, the concentration of the stressor, and the duration of the stress signal that's going to determine whether or not you get immunosuppression or just immune relocation during a stressful event. We also have this thing that is relatively new in the past couple thousand years, which is chronic stress. Historically, we had more acute stress. Now we're experiencing more chronic stress. Our stressors don't go away. When we're discussing stress, we're particularly talking about these three cytokines: IL-1, IL-6, and TNF-alpha. And the degree of pathology that we see with stress is associated with the amounts of these three cytokines. Now, whether you have an acute or a chronic stressor or acute or chronic inflammation is dependent on what you're experiencing. So an infection is typically an acute stressor, and we'll see a high level of IL-1, IL-6, and TNF-alpha. But we get chronic levels of those three cytokines from a long-term stressor, from hypersensitivity reactions. You could think about food hypersensitivities, that people eat those foods every day, not aware that they are hypersensitive to them. Or you could think about hypersensitivity reactions that people are having to environmental chemicals. So we're starting to see more hypersensitivity reactions to things like laundry detergent, shampoo, the metals that are in our clothing and whatnot. We know that dysbiosis is leading to chronic inflammation for reasons we've already discussed. Obesity leads to chronic inflammation, and obesity is interesting because we see that in fat, macrophages will take up residence, and those macrophages will chronically produce IL-1, IL-6, and TNF-alpha. So just by losing weight, you reduce chronic inflammation. And then one of the number one causes of chronic inflammation is gum disease. And we see that specifically with cardiovascular disease. We can link a lot of cardiovascular inflammation back to periodontitis and gum disease. What's important to remember about these cytokines is that they do not only lead to an immune response, they also have behavioral patterns that are associated with them. So interleukin 1 causes malaise, fatigue, and depression. Now, what's interesting about this is that it is a bidirectional relationship. So if you are fatigued or depressed, you will produce IL-1. And in some people, we can't tell the chicken or the egg. We can't tell, did they have a chronic inflammatory reaction happening that led to the production of IL-1 and now they are fatigued? And for other people, did they experience a loss of a loved one and that made them depressed and now they are producing IL-1? But in the case of COVID, we know the infection that led to the production of IL-1. So when we see people with post-COVID and extreme amounts of fatigue, we can make the leap that IL-1 is likely there and responsible for the hypothalamus causing us to feel tired and wanting to go to bed. IL-6 is the second pro-inflammatory cytokine, and it's associated with fatigue as well, but also anxiety. So when we're stressed and we're feeling like, oh my God, I can't get sick today, that's IL-6. IL-6 is also bidirectional. We see a high level of IL-6 in people who are catastrophizers, and they often show up in pain clinics. And then TNF-alpha is associated with aggressiveness, hostility, but it's also associated with pain. What's interesting about TNF-alpha is that when we were studying the people with inflammation from cardiovascular disease, we saw much higher levels of TNF-alpha. And if you go back and look at the populations, these were primarily people like stockbrokers. We've also studied people who are athletes, who are in competition on a regular basis, and they tend to produce higher levels of TNF-alpha. Interestingly, athletes will produce TNF-alpha up to 2 hours before they have a match. So it's like their body is preparing for injury. Now, the biochemistry of how these cytokines are having an effect on our emotional and behavioral state is kind of interesting. It goes back to the kynurenine pathway. You'll remember that the kynurenine pathway happens when you ingest protein, and then tryptophan from that protein is typically converted by your gut microbiome into serotonin and tryptamine. But if you have inflammation, tryptophan is shuttled through the kynurenine pathway instead of producing serotonin and tryptamine. And I just want to point out that just past serotonin, if we extended out this pathway, would be melatonin. So this is also why people can't sleep, because tryptophan, instead of being shuttled to serotonin and melatonin, is shuttled through the kynurenine pathway, where we get the production of quinolones, and those quinolones are neurotoxic, causing the brain fog and the fatigue that we experience when we have inflammation. So if we now go back and look at some of the symptoms of long COVID, we recognize that that feeling tired and malaised can be the production of IL-1 and IL-6. Those inflammatory cytokines cause long-term fatigue. So addressing post-COVID in patients needs to also address chronic inflammation. And this may be chronic inflammation that people had before they experienced COVID, but COVID seems to exacerbate whatever underlying condition people have. So here's where we are so far. We're recognizing that underlying inflammation can lead to those feelings of depression, anxiety, hostility, and aggression. And if someone has depression, anxiety, or aggression, they also likely have an overproduction of the inflammatory cytokines. I want to bring in social support because social isolation has been a real reality, and it absolutely has an effect on our immune response. So we have been isolated, and I thought this was the best illustration of how I have been feeling, and I think a lot of people have been feeling since the pandemic. What is isolation doing? Well, isolation means that we're less exposed to other people's microbes. One of the reasons we've had less infections over the last 2 years. We've also been wearing masks and using hand sanitizer. And what that does is it means that we are too clean. We are not getting exposed to microbes. Hence, we are not making interferon gamma. We are not getting infections. This is a problem for us. We're also seeing less exercise and people overeating. And so here's our chronic inflammation that's coming from fat. Less exercise means that the metabolites that our microbiome makes aren't getting pushed into the bloodstream. And so even if we're eating 100% perfect, if we're not getting exercise, all of our organ systems are not receiving those metabolites. We also have the stress of media and politics, and all of those things feed into an imbalance that we are experiencing of microbial metabolites, neurotransmitters, cytokines, hormones, the endocannabinoid system, the opioid system. They're all out of balance. I love this experiment. It was done in animals, but I still think that this is such an important thing. The first part of the experiment was in humans, where they were showing social reward in complex environments, and a smiling face means positive social reward. Like, we experience true social reward just from someone smiling at us. Now imagine 2 years, well, you don't have to imagine, you experience 2 years of people wearing masks where you couldn't see them smile. What a smiling face does is it triggers the same dopamine pathways as getting a monetary windfall, or eating your favorite food, or psychostimulants. And the psychostimulants is what they did in animals to show that it was the exact same behavioral pathway. And this is the dopamine reward pathway. So let's just recall that dopamine, serotonin, GABA, acetylcholine, and probably glutamate are all interacting to produce our mood states. Dopamine is our reward neurotransmitter. Serotonin brings us happiness and joy. GABA is our relaxation neurotransmitter. We make most of our GABA at night when we're sleeping. So if we're not sleeping, we're not making GABA. And then acetylcholine keeps us calm. But we're now recognizing that these neurotransmitters are not just being made in the brain. They are also being made in the gut. And in fact, serotonin is probably 95% made in the gut. So if we think about these moods, dopamine is our reward neurotransmitter. That's going to be the neurotransmitter we make when we take out our winter coats and find an extra $20 bill in there. And we're like, woohoo, right? But if you look in your wallet and you see $20 and you knew you went to the ATM, no reward. So it has to be unexpected reward for dopamine. It also gives you a positive affect and makes you more extroverted. Serotonin is for happiness, motivation, and calm. GABA, relaxation, focus, and sleep. And it's our inhibitory neurotransmitter for dopamine. And acetylcholine is for calm and anxiety regulation. Doctors Data has a comprehensive neurotransmitter panel. So if you have questions about whether your patients are making enough of these neurotransmitters, you can actually run this panel and it will tell you serotonin levels, it will tell you glutamate levels and histamine, as well as your dopamine and your GABA. So highly recommend this for folks who are struggling with moods and coming out of social isolation. But it's also important for us to remember that here comes our microbes again. Not only are they modulating our immune system, they're also modulating our nervous system. So GABA, relaxation, is produced by lactobacillus and by phytobacterium, not stimulated by, produced by. These microbes can make GABA. Likewise, we see serotonin from Candida, Streptococcus, E. coli, Enterococcus, dopamine from Bacillus, acetylcholine from Lactobacillus. If we have dysbiosis, the likelihood that we have a dysfunctioning neurotransmitter system is very high. So here's where we are. Our microbes are also controlling our neurotransmitters. And don't forget, we can get reward. We get a dopamine burst from just smiling. So I know you do this with babies. I know you go up to them and you give them a smile, but maybe you can do it with, everyone. And we would start fixing that neurotransmitter system. Our social isolation will have disrupted our neurotransmitters, our hormones, and our microbial metabolites, leading to immune dysfunction. And there's a lot of power, even smiling at people we don't agree with. So this should tell you how I suggest then approaching bringing the immune system back into balance. We want to lower our baseline inflammation. We want to make our specific immune system able to respond quickly to an infection and then go back to normal. None of these prolonged responses, none of these things where we get stuck in specific immunity and we don't resolve. We want to be able to resolve inflammation, resolve our infections, and prevent autoimmunity. And in order to do that, we have to really focus on these three components of the immune system. You'll notice the innate immune system is not highlighted here. And that is because our innate immune system has to function or we die. Our ability to change our innate immunity is relatively limited simply because if your innate immune system isn't working, you're not going to survive. It's these parts that are more malleable and that we can pretty much change based off of our lifestyle and our behavior. The way that we do that is we support a healthy T regulatory population. So for those of you who are less familiar with T regulatory cells, these are cells of your immune system that are found in your gut that produce TGF beta, transforming growth factor beta. And TGF beta can regulate interferon gamma. It can control the levels of how much interferon gamma we're making. It can regulate our allergies by regulating IL-4, IL-5, and IL-13. It can regulate asthma by controlling IL-9, which is the cytokine that actually remodels the lungs in asthma, and IL-10. And it can control our response to mold and our autoimmune reaction by controlling IL-17. It really is one of the key cytokines that our immune system makes. And again, it's made in the gut. So if we have happy T regulatory cells, that means we have a happy microbiota and a healthy immune system. Likewise, a happy microbiota can give us happy T regulatory cells. So if we focus here, we're kind of going to have an effect everywhere. And how do we get healthy T regulatory cells? Well, we get it through diet and nutrition. That is the number one place where we're going to get our healthy Tregs. So let's go back to our normal immune response. Which nutrients do we focus on for healthy Tregs? And let's start with vitamins. We know that vitamin A, and specifically retinol, is going to lead to production of healthy T regulatory cells. Specifically, when we're worried about autoimmunity, vitamin A has been studied in animal models of just about every autoimmune disease out there. It has not been studied in human autoimmunity as much, but the animal studies are very clear. It's really good at producing healthy T regulatory cells to shut down autoimmunity. Vitamin D helps us fight infection and control inflammation. And vitamin E also has an effect on inflammation, but its real role is over here at specific immunity. Vitamin E keeps our T cells healthy, doesn't skew them to be Th1 or Th2 or Treg. It just keeps their membranes healthy, keeps the T cell receptor healthy. So, when we're looking for healthy levels of T cells, vitamin E is where we're going to go. Vitamin A specifically for Tregs, vitamin D also for autoimmunity, but even more for infection. When we're looking at resolving infection, that's where we look at our omega-3 fatty acids. Resolution and getting inflammation resolved is coming from omega-3s. So how are you going to assess where your patients are with these particular nutrients. And Doctors Data has a fat-soluble vitamins serum test and a fatty acids test that will test the omega-3 fatty acids in the blood and the fat-soluble vitamins. Let's go back to why those omega-3s are so important. The omega-3s are involved in producing some anti-inflammatory lipids. So you will recall from when you went to medical school that omega-6s are involved in producing arachidonic acid, which gives us our pro-inflammatory lipids, the leukotrienes and the prostaglandins. What's been discovered, probably since you went to medical school, is that the omega-3 fatty acids are responsible for producing the anti-inflammatory lipids, specifically the resolvins, the protectins, and the maresins. Now, where do we get omega-3 fatty acids? We get them from fish oil, or we take them specifically as omega-3s. You can get omega-3s from flax oil. They aren't in the same ratio as they are in fish oil. But how many Americans are getting enough omega-3s? Not very many. So if we look at why we have such high levels of baseline inflammation, it is likely the lack of omega-3 fatty acids, as opposed to there may be an overingestion of trans fats, but even more important would be the underingestion of omega-3s. How else can we get TGF-beta from our T regulatory cells? Well, we know that nutrition, and specifically prebiotic foods, will do it. When I'm saying prebiotics, I'm specifically talking about the foods that have non-digestible fibers and inulins. These are going to be things like Jerusalem artichoke and leeks and onions and bok choy. Some of these fibers are fantastic for increasing our microbes. And that's the next piece we look at. We look at our microbiota, and in particular, our bifidobacteria are what are giving us a healthy T regulatory population on a consistent basis. So if you're looking at a probiotic, you're looking specifically at a probiotic that contains bifidobacteria. And there are multiple strains, so usually you'll see it as B. breve or B. adolescentis, but you're looking for the bifidobacteria. And then vitamin A. And if you read the literature, you think, oh, well, everybody gets appropriate levels of vitamin A because you get them from carrots and you get it from oranges. But what we're seeing actually is that the varietals of oranges have changed over the years. And we now get oranges that are the size of a grapefruit, but have minimal amounts of vitamin A. They're mostly water. And what's been shown is that you would have to eat 20 oranges today to get the amount of vitamin A that you got in a single orange 20 years ago. So is it possible that people are low in their vitamin A? It is. It's something to start measuring. As I mentioned, with prebiotics, we're looking at the ingestion of dietary fiber. And then that dietary fiber gets fermented by our bacteria or our probiotics. And then that fermentation leads to the production of short-chain fatty acids and other microbial metabolites, which are called postbiotics. So we've got our prebiotics, our probiotics, and our postbiotics. And that means that if you feed your microbiome, those microbes are going to make the metabolites, and the metabolites are going to be what impacts the immune response. And this is where we have to remember that our microbiomes are ecosystems, and mine is not like yours. It's more than just the presence or absence of microbes. Some microbes are more important than others. And we have to remember that microbes are highly sensitive to their environment. They are the so-called canary in the coal mine. The microbes are going to die before you do. They're going to alert you that something's wrong. They can be killed by all sorts of different chemicals, as well as preservatives in foods and different medications. And the health of that microbiome influences our immune system. So we have to pay attention to all of these aspects. Now, I mentioned prebiotics, probiotics, and postbiotics, but I want to go into a little bit more detail here for a minute and just remind you that Probiotics, most probiotics are live bacteria when they are made, but by the time they reach your gut, most of them are not alive. And in fact, what we think is causing the health benefit from a probiotic are postbiotics that are in the bottle. So if you put a live bacteria in a bottle with food, it's going to produce metabolites or postbiotics. And we think that is actually what's causing the benefit that we see with a probiotic. We also have a new type of probiotic out that's called a parabiotic. This is a heat-killed probiotic, and these are now being found in foods. Why? Because it's way easier to administer a parabiotic that you don't have to keep alive. People have been trying to put probiotics into things like orange juice for a long time, and it just kills the probiotics. And now by using heat-killed probiotics, now they're able to do this, and they still work. So the fact that they are heat-killed and they work tells us that it's the postbiotics, it's the metabolites that are actually having the effect. It's not the live bacteria. And then there's synbiotics. Synbiotics are encompassed by fermented foods. This is our probiotic plus a prebiotic. So as you move into the future, you are going to see more and more references to postbiotics. I recently saw an antiperspirant that has postbiotics in it, and parabiotics. And again, the idea here is that you administer the metabolites instead of the live microbes, and it's considered less risky and more healthy. We know more about what we are doing when we administer a postbiotic than when we administer a probiotic. The way that we measure microbiome health is with alpha diversity, and that GI 360 test that Doctors Data does measures this. And alpha diversity is the diversity of microbes within one person. We can also measure beta diversity. This is when we're comparing two people. So if I'm comparing the diversity of my microbiome to the diversity of your microbiome, that's beta diversity. And the other thing that we measure is a Firmicutes Bacteroides ratio. What we tend to see is that as Firmicutes increase, health decreases. So you want less Firmicutes, more Bacteroides. You are looking at people who are unhealthy, they're going to have more Firmicutes and less Bacteroides. And we know that loss of diversity is bad. This is a study that had 30,000 people, so large study, and you're going to see enormous p-values like p-value of 10 to the minus 122. Never seen a p-value that big before. But what we see here is that as BMI increases, here we see alpha diversity decreases. As hemoglobin A1C increases, alpha diversity decreases. As blood glucose increases, alpha diversity decreases. So when you have dysbiosis, you're seeing all of these health measures get worse. What that means is that if we can address this unhealthy microbiome, it will have an effect on the metabolites that those microbes make. And that may allow us to then rejuvenate our immune system, improve our response to infection, improve aging and inflammaging, decrease autoimmunity, and decrease neurological disease. So what can you do today with you or your patient? You can increase your dietary fiber, and that's going to increase the microbial diversity. You can increase your microbial diversity, which is going to increase the production of short-chain fatty acids, and that's going to cause TGF-beta production and immune health. And I love this study by the American Gut Project. They were looking to see whether or not you could get better alpha diversity with an omnivore diet, a vegetarian diet, no red meat, eat seafood, or vegan. And what you can see is all the colors are in both places. And what was different about this group over here on the left, which has less diversity, and this group over here on the right, which has more diversity, is that this group ate 30 types of plant-based foods per week. Let me repeat that. 30 types of plant-based foods per week. Not 30 servings, it's the diversity. It's 30 types of plant-based foods per week. So what I'm encouraging you to do is get your 30. Mix up the plant-based foods you get. Remember, spices count. Remember, nuts count. Beans count. 30 plant-based foods per week. What we see with the average American is they're under 10. So gradually working them up to 30 plant-based foods per week is going to rejuvenate their immune system. As we improve our lifestyle measures, it is going to allow us to improve all of the underlying proteins and endocannabinoids and hormones and everything. So start with the 30 plant-based foods per week. Try to get rid of some of the stressors. And maybe that involves turning off the media or meditating or finding good coping behaviors. We need movement to get those metabolites into our bloodstream. So get up from your computer, go outside, and find excuses to move. We also need sleep because that GABA that we make in the middle of the night is decreasing our inflammation. So we really want to sleep through the night. We need to get that microbiome healthy and diversify it. And the way to do that is with gardening. Soil-based microbes are actually really good for you. Having pets, going into nature. We also need less hand sanitizer, less antibiotics, less non-antibiotic medications, less pesticides, and less preservatives, all the things that kill our microbiome. We should be looking at whether or not we're getting prebiotics, probiotics, symbiotics, and fermented foods in our diet, and be considering a postbiotic if we're in a state where we might have COVID or long COVID. Our immune systems have suffered, and as a result, we have chronic inflammation, which is contributing to depression and anxiety. Our lack of social interaction has decreased our dopamine, oxytocin, and our immunity. And so to address immune rejuvenation, we got to address that microbiome, got to go to the gut. The central focus, increase the microbial diversity, eat 30 plant-based foods per week. There you go. This is how you find me. And I will now answer some questions.